You know that feeling in the pit of your stomach when you check your flask and something is off? That cloudiness, that fuzzy ring, these are the microscopic contaminants that are, and I'm not kidding, the stuff of every cell biologist's nightmares. First up, you've got the most obvious threat, fungal contamination. Think of this one as the brute force attacker. It shows up looking all fuzzy, kind of like web-like filament, or it just makes everything cloudy. And it's spores. These spores can float through the air looking for any tiny weakness in the defenses to establish themselves. Next, we have the rapid opportunist bacteria. And this one moves fast. A culture can look perfectly fine 1 minute, and then just a few hours later, the media, that's the nutrient soup the cells live in, is cloudy and has totally changed color. Bacteria multiply so quickly they can just overwhelm an entire experiment before you even realize what's happening. And then there's the most dangerous one of all, the invisible saboteur, Mycoplasma. This is the ultimate threat. This tiny bacterium is so small, it doesn't make the media cloudy. It gives you zero visual clues. It just quietly gets in, starts messing with how the cells behave, and silently renders months or even years of research completely useless. So how do you defend against these infiltrators? There are many parts of a cell culture space that need to be thoroughly cleaned regularly, routinely, and especially when there's contamination. You have your incubator, you may also have a water bath, you may have a clean bench, or more suitably for culturing any kind of mammalian or human cell culture. So if you suspect or you see any of these contaminants, the first step is mission critical, and that's containment. The second a breach is spotted, everything stops. This step is all about making a bad situation not worse. You stop and you immediately label the contaminated flasks or dishes. Never open a contaminated flask inside the tissue culture hood. Wipe the outside with bleach, then send the sealed biohazard bag directly to be sterilized in an autoclave. The next step, we need to decontaminate the incubator. This is where you put your cells, the home for the cells. The incubator is warm, humid, and may be full of CO2 for mammalian cells, for human tissues. And this is perfect for cells, but also perfect for contaminants. You have to nuke this space. The first step is to empty it out, remove all shelves, trays, and the water pan. Disassemble. Take apart any removable internal components. Drain the water pan and sterilize its contents before disposal. Unleash the bleach. Meticulously wipe down every single internal surface, so the walls, the ceiling, the floor, the gasket, with a fresh 10% bleach solution. The 20-minute rule. This is the most crucial. The bleach must sit on every surface for a full 20 minutes. This non-negotiable contact time is scientifically proven to be the minimum required to kill the hardest fungal and bacterial contaminants. Then you can rinse and reassemble. Wipe surfaces with sterile distilled water to remove corrosive bleach residue before putting the parts back and turning the incubator back on. Step 3, we want to secure the sterile workspace, the hood. So your Biosafety Cabinet or clean bench is the frontline defense. It needs the same precise multi-step treatment. Remember, if you're working with anything that could harm you, like Plasmodium falciparum, you must be in a Biosafety Cabinet, which protects both you and the cells, not just a Laminar Flow Bench, which only protects the cells. So what you do is do an initial wipe with 10% bleach. You want to saturate and allow contact time, minimum 10 minutes, up to 20 minutes or longer to kill surface contaminants. Then there's a second wipe, and this is the rinse. You rinse with sterile distilled water, remove the corrosive bleach residue by using sterile distilled water. Then the third wipe here, we're polishing. So 70% ethanol, final clean, ensuring a completely sterile and non-corrosive working surface. So the final test, we trust, but we verify. You don't just assume the counter operation worked. You run a test. You take a flask of perfectly sterile, clean media, which is the nutrient soup. You place it inside the decontaminated incubator for a few days. If the media stays crystal clear, the vault is secure. If it gets cloudy, you missed something and the protocol starts all over again. You may have to autoclave the bits and pieces that you removed from your autoclave. But remember that the breach protocol that we just went through is the last result. The real goal is to stop a breach from ever happening. That means shifting your entire mindset from reactive cleanup to proactive, relentless prevention through rigorous daily, weekly, and annual maintenance. So that brings us to fortifying the defenses. You've heard the saying, right? In the lab, it's 100% true. The best defense is a good offense. And this is exactly what the offense looks like. It's a rigorous, non-stop schedule of maintenance. Every single day, the bench gets wiped. Every single week, the incubator water is changed. Every month, a full deep clean is performed. And every year, a certified technician comes to make sure the HEPA filters, the actual lungs of the Biosafety Cabinet, are working perfectly.